Hello, everyone, and welcome to another video on Our Own Devices. I'm Jean Messier, and today I'm starting a multi-part series on Canadian and British military NBCW or CBRN equipment. So NBCW stands for Nuclear, Biological, and Chemical Warfare or weapons, while CBRN, the currently preferred term, stands for Chemical, Biological, Radiological, and Nuclear, with the radiological term being added to cover dirty bombs, that is, conventional explosives salted with radioactive elements in order to contaminate the target area. So the equipment we're going to be looking at in this series is carried and worn by soldiers in the field in order to protect them from said weapons. Now, I thought that I would start off this series with a follow-up to a previous video. So you'll recall that a little while ago, I did a video covering chemical warfare agent detection kits, specifically the L1A1 kit from the 1950s and the C2 kit from the 1970s. Well, we're going to continue that series with the M256A1 kit. So in my previous video, I noted that the older detector sets tended to be pretty complex and cumbersome and really fiddly to use, especially if you were wearing bulky protective gear. And indeed, the US military thought so too, which is why in 1971, they put out a requirement for two new chemical detector sets designated the XM189 and the XM190. Now, the XM189 was to replace the older AN-M15A2 detector kit, would be used to detect nerve agents and blister agents, while the XM190 would replace the AN-M18A2 detector kit and would be used to detect arsenicals, phosgene oxime, and blood agents. However, in 1972, this requirement was changed to a single kit capable of detecting all of these chemical warfare agents, and the development contract was awarded to the Environmental and Process Instruments Division of the Bendix Corporation, and the result was the M256 kit, which was given its type designation in 1977 and first widely issued in 1978. Now, this particular example here is a Canadian version manufactured by Anachemia Canada Incorporated out of Lachine, Quebec. The whole kit is contained in a rigid plastic box and includes a strap so that it can be hung from the user's neck, leaving their hands free to manipulate the contents. Now, if we open this up, we'll see that the contents are a lot less complicated than the previous kits that we've covered. So this would originally have contained 12 individually packaged M256 sampler kits, a pair of plastic instruction cards tethered to the box by string in both English and French, because Canada, and a booklet of M9 three-way detector paper, and this would turn orange in the presence of G nerve agents, dark green in the presence of V nerve agents, and pink or red in the presence of blister agents. However, this paper was only designed to detect liquid CW agents. It couldn't actually detect vapors in the air. For that, you used the M256 sampler kits. Right, so if we pull this out of its packaging, which, by the way, contains another set of instructions in case you lose the original box and instruction booklets, we can see that this consists of a small plastic card with a number of reaction spots for detecting different types of chemical weapons. These are made out of various absorbent materials, and the reagents are contained in little glass ampules. And the idea is that you would break these ampules with your thumbs, and the reagent would be released and be absorbed into these reaction spots where they would react with any chemical warfare agents in the atmosphere to produce a color change. So this is designed to detect 5 different types of chemical warfare agents. It can detect nerve agents, both G-type, such as sarin and tabun, and V-type, such as VX. It can detect mustard gas, both H and HD type, and mustard gas is a blister agent or vesicant that inflicts severe burns to any exposed tissue. It can detect lewisite, which is another type of blister agent based on arsenic rather than sulfur. It can detect phosgene oxime, which is a separate type of vesicant known as a nettle agent or urticant. And finally, it can detect blood agents, specifically hydrogen cyanide and cyanogen chloride, which interfere with the body's ability to absorb oxygen. Right, so let's have a closer look at this and see how it's actually meant to be used. So the first step is to peel away this tab to reveal a lewisite detection tablet or crayon. You then fold over this tab and swipe it across the crayon, leaving a pale brown streak. And this crayon contains Michler's thioketone, or 4,4-bis-diamethylaminothiobenzophenone, which turns dark green on exposure to lewisite. And for now, that's all you need to do. We'll actually return to this step at the end of the entire process. Now, the next step is to break all of the glass ampules marked with A3. And you would do this with your thumbs and preferably using this little square of blotting paper. And this is to prevent the glass from the broken ampules from penetrating your protective gloves, which if that happens, you're going to have a bad day. And this is going to release the reagent. And what you're supposed to do is hold the card upside down to allow the reagent to flow into and fully saturate the reaction spots. So the center circular spot is used for detecting blood agents and is made out of fiberglass chromatography substrate treated with barbituric acid. And one reagent ampule contains calcium hypochlorite, while the other contains 4-benzylpyridine in an ethanol solution. And what happens is that the calcium hypochlorite reacts with hydrogen cyanide to produce cyanogen chloride, which then reacts with the reagent to produce either a pinkish red or pale blue color. Right, so if we move over to the square reaction pad, this is made out of silica gel and is intended to detect mustard gas and phosgene oxide. One reagent ampule contains 4,4-nitrobenzylpyridine and mercuric chloride in a methanol solution, while the other contains potassium carbonate. Now, for maximum sensitivity, this reaction has to occur at high temperatures, and this is accomplished using this little chemical heater. So this consists of 2 ampules full of cupric chloride and a disc of sintered aluminum powder, and when these two mix together, they undergo an exothermic reaction. So how you carry out this test is you swing away the heater, you crush one of the ampules, not with the blotter paper this time because you don't want to crush both at the same time, and then you swing it back into place and wait for 10 minutes. Then once your 10 minutes are up, you swing it away, crush the ampule labeled 5, crush the second cupric chloride ampule, swing the heater back into place, and wait another 10 minutes. And once the reaction is complete, the reaction spot will turn blue in the presence of mustard gas and red in the presence of phosgene oxide. So the star-shaped reaction pad is for detecting nerve agents and consists of filter paper impregnated with cholinesterase enzyme. Now, if you want to learn more about nerve gas, how it was developed, and how it works, please check out the video I wrote for Today I Found Out on the subject, link in the description. But briefly, nerve impulses are carried across the synapses between different neurons or between neurons and organs or muscles by neurotransmitters such as acetylcholine. However, once the impulse has been sent, the acetylcholine needs to be broken apart by cholinesterase enzyme and reabsorbed by the neurons so that the synapse is reset and another signal can be sent. And this process is known as reuptake. Nerve agents, however, interfere with the action of cholinesterase, meaning that acetylcholine just builds up and the nerve starts firing continuously. If this happens all over the body, the result is convulsions, paralysis, and a swift death. Now, interestingly, the first M256 kits used cholinesterase enzyme derived from horse serum, but this was found to be insufficiently sensitive to protect troops against VX gas. And so it was replaced with cholinesterase enzyme derived from electric eels, which was 10 times more sensitive. And these upgraded kits were designated M256A1 and were first widely issued in 1987. Right, so the 2 reagent ampules for the nerve gas test contain a buffer solution and a solution of 2,6-dichloroindophenyl acetate in petroleum ether, which turns blue when it reacts with cholinesterase. However, if nerve agents are present, they will inhibit the action of the cholinesterase, meaning that this reaction can't take place. So, unlike with the other spots where a change in color indicates a positive result, here a change in color indicates a negative result. So if the color doesn't change, that means there is nerve agent in the air. And finally, we come back to our lewisite test. And what you're supposed to do here is fold this tab back down and make another streak on the lower paper using the crayon. And the idea here is that in the time it takes to carry out the rest of the steps in this procedure, the first streak has been reacting with any lewisite that happens to be in the environment. And the second streak acts as a point of comparison. So if the first streak has significantly darkened or turned green, that means lewisite is present. Now, while this is certainly more compact and straightforward and has fewer small parts that you can lose than earlier chemical detection kits, To my mind, this would still be pretty fiddly and complicated to use in the field, especially wearing heavy protective equipment. Though I suppose the user-friendliness of these kits is somewhat limited by the complex chemistry involved. Now, since it would be a rather bad idea to train troops using actual chemical warfare agents, training versions of these are available that undergo the same color changes without needing to react to actual poison gas. These are the M28, which simulates the detection of nerve agents, and the M29, which simulates the detection of blood agents, blister agents, and phosgene oxime. Now, it's important to point out here that the M256 kit is not intended for use as a CW agent alarm. It's far too slow and cumbersome to use to provide any sort of early warning. Rather, this is intended to confirm the presence, the identity, and the concentration of CW agents in an area where their presence is already suspected, and to indicate whether it is safe for troops to remove their protective equipment or whether a particular area is safe to traverse or make camp in, for example. And while kits like this are still widely issued and very useful, they are being increasingly supplemented by electronic detectors such as the Chemical Agent Monitor, or CAM, which uses ion mobility spectrometry to achieve the same results far more accurately and much faster, on the order of seconds. Though that, I think, warrants its own video, which I will produce as soon as I get my hands on one. So stay tuned for that. Anyway, that's all I have for you today. Thank you so much for watching, and I'll see you in part 2, where we'll start looking at CBRN protective equipment, starting with post-World War II Canadian gas masks. Until then, I'm Jean Messier from Our Own Devices. Have a great day.